been blessed to be active in the field of decomposition ecology since 1994. As my children like to point out, that was the last century. I've been really active in that area, and I'm going to give a general presentation that's going to pivot a little bit from the topic. I'm going to approach it more from a decomposition ecology perspective, generalists and specialists, and how they compete for resources in nature, and what does that mean for nutrient recycling, and then how do we apply that work, obviously, in the insects as food and feed industry. Let me share my slides. All right. Everybody see my slides?
Yes.
Excellent, excellent. So the title that I modified it a little bit. I'm going to focus on micronutrient concentration and the impact on the early stage development of Hermetia lucens, the black soldier and Cochleomya macellaria. It's called the secondary screwworm. It's in the family Calliphoridae, and talk about the implications for insects as food and feed, and maybe a little bit about forensics. I do want to point out that what I'm presenting is really an emphasis on work that Erin Harris conducted for her PhD in my lab. She now works with Tyson Food Group. So I just want to shout out to her for her effort in developing these slides and her generating the data that will be presented. And then at the end, I'll swing over and talk a little bit about Tara's work and what she's doing. So for me, in terms of the research I do, I really focus more on systems ecology. And when we think about nutrient cycling, there's two primary areas where we think about production and then breakdown. And the first, obviously, is the green food web, and we think about anything that uses the sun to generate organic matter. And that's what happens above ground. And we think about the sun impacting plant growth, and that leads to animals that feed on those plants, etc. But when we think about the brown matter food web, we're talking about the breakdown of these materials. So we have this push-pull system of production and then degradation and recycling. And we know with nature, nature's very conservative. Everything's going to be utilized efficiently. So when we think about decomposition ecology, decomposition is the breakdown of organic matter into gases, water, nutrients, and energy, which are then recycled within that given ecosystem. Uh, Detritus, which is autotrophically derived, is decomposing organic or plant matter. So this is something that can be continuously produced throughout the year, depending on the location. But then there are heterotrophic systems. So we think about carrion production, vertebrate decomposing remains, and how that can serve as a resource pulse into a given landscape. We published this work. Eric Bimbo and I and several others put this together into what we call the necrobiome. So my lab, I'm probably known more for black soldier fly work, really focuses on this system and what regulates it. So we think about um, what materials being produced and how it's being decomposed, but also how species interact with one another in this given landscape. Now, originally, as pointed out here, it was about heterotrophic biomass, but we've decided that the autotrophic systems were just as important in this process because we know it's a bottom-up driven system of plant production drives the heterotrophic system on top. So when we think about heterotrophic and autotrophic, I mean, concepts that we really like to investigate in my lab are topics like succession or coexistence, niche partitioning, metapopulations, especially because when you think about heterotrophic systems, predictability of carrying in environments can be challenging in some circumstances, but in others, maybe not. So we think about salmon runs in rivers or mass cicada emergences in the United States. These are predictable pulses into a given ecosystem. So there are a lot of factors, obviously, that impact these processes. We think about abiotic factors such as temperature, the sun, rainfall, etc. But there's also going to be biotic factors that play a role, from the microbe up to insects to even vertebrates that scavenge for these resources in a given landscape. What's really fascinating about this, and Erin really emphasized this with her PhD work, was that we see a lot of variation in these ecosystems. And a key question is what drives this variation, and why in some circumstances do we see little variation, but in others we see large variation? And that'll be relevant down the road when we talk about what does this mean for mass production of insects. Variation can kill a business model. So, how do you control it? So, a lot of the work that we'll discuss moving forward will really focus on how nutrient manipulations can impact the variation observed in a production system. So why does variation exist? Well, there's a lot of factors, obviously, that play a role in it, from genetics to the scale that we're doing the work and the density of the larvae that we're working at, the temperatures that are experienced either within the environment, the enclosure, the facility, or even within the pan, down to moisture content of the resource, nutrient concentration, and even competition, which may not be something that we want to see in the uh, facility, but we obviously recognize that intraspecific competition is really important for these processes. Now, when we think about nutrient variation, um, it's fascinating to look at what's in the literature today with black soldier fly, but if we expand out and just think about uh, our nutrient availability in the ecosystem, this gives you this is an image from a 2019 paper that I published with Eric, and it shows you the volume of material that's produced for these different categories. So you think about carrion here or wood production systems here, and this gives you the carbon nitrogen ratio, but the volume of material that's being produced. Historically, most efforts really focus on the green food web, so the autotrophic driven systems. Very little is known about heterotrophic systems. And again, that's where my lab spends a lot of focus. What's fascinating is when we look at this carbon nitrogen ratio, is the variation observed in that. So carrion's much tighter in terms of variation versus wood. And why is that? Well, I think that's because we're talking about a much more diverse world from the plant perspective than the vertebrate system or the heterotrophic system. When we think about nutrients and work that's been done over the years, macronutrients are really important. We see a lot of papers that are on lipids, carbohydrates, etc., and they're all treated equally. So with protein, we think about what's produced in like meat-based products. It could be plant-based as well. So we think about legumes that can have a lot of protein, carbohydrates, so that could be your sugars, and then lipids, which would be your fats. But when we think about research that's being done, they're treated equally in terms of formulations and feed production for black soldier fly. My lab is really starting to focus on micronutrients. So we're very interested in how vitamins, minerals, and even specific amino acids can serve as bottlenecks in ecosystem. Now that could be outdoors, but it could also be in mass production systems. One single micronutrient could serve as a bottleneck regulating that whole process. And we're kind of looking for the needle in the haystack to know which one or group is playing the major role in that process. Because if we can manipulate those micronutrients, we can unlock the capability of the insect to recycle those materials quicker, more efficiently, etc. So the micronutrients that we focus on right now in the lab, we're doing quite a bit of work. And again, I'm focusing on Aaron's PhD work. We're focusing on tryptophan. It's an essential amino acid. It's an important amino acid for hormone synthesis, metabolism, locomotion, stress, immunity, or stress response. And then we're also looking at zinc. Zinc is an essential trace element, it's a cofactor for a number of enzymes. So they both play a role. And what's really interesting is they interact with one another. And one thing on tryptophan that I've written a little bit on is this idea that tryptophan also is a key amino acid that produces many quorum sensing molecules. And the one that I've spent most time working on is indole. Indole is a derivative of tryptophan, and that regulates a lot of microbe-microbe interactions, but also microbe-host interactions. So while I'm talking today about the insect perspective, a layer to this is understanding how the microbiome is responding to these compounds as well. So I think going back to this process, the microbiome, is talked in a very general sense. What we really want to focus on is this nutrient availability and the digestibility, because just because it's there does not mean that it's available for the animal to consume and utilize. Okay, so as I shift, this is also a major part of research in my lab, and we try to approach it. There's two facets. One is autotrophic versus heterotrophic systems. What ecological parameters are similar across systems and where do they differ? but also looking at it from a generalist versus a specialist perspective of what's consuming this material, where you may have a caterpillar that feeds on one or two plants or specializes on feeding on a lot of different plants. So this is also from the green matter food web, but we see this a lot. And this is a major area of focus in row crop production, et cetera, of when you deal with pest species, are they generalists or are they specialists? And it's also involved when you think about trophic interactions with parasitoids and what will they colonize and how I can use that in integrated pest management, et cetera. But this nutritional plasticity across all of these resources really does impact degree of specialization. Where do you want to put all of your effort? Do you want to specialize on one particular niche and be the best at it and risk that resource not being available? Or do you want to be a generalist where you can feed on a lot of different things, but maybe not be as competitive? So there are trade-offs depending on what strategy is selected. And I say that in a very general term, selected from an evolutionary perspective for specialization. So when we look at this, when we think about detritus and carrion, we know that some animals will focus specifically on plant-based matter, but there are those that will feed on both. There are animals that will feed on plant-based matter and on animal-derived matter, so autotrophic and heterotrophic systems. So Macellaria in our world is considered a specialist. This is Cochlearia macellaria, and I'll talk about it in a minute. It is a specialist. It colonizes carrion. But Hermetia lucens is a generalist. It can feed on a lot of different resources. And I just want to point this out. I'll talk about their biology in a minute, but what's really fascinating about it is their evolutionary stable strategies. If you look at Macellaria, Macellaria can live a long time. It feeds actively as an adult. It can mate multiple times. It lays multiple clutches of eggs. But the trade-off is it only colonizes carrion. Now, look at the black soldier fly. Short life cycle, one, maybe two clutches of eggs, very small number of oviposition events. They they don't have to feed as an adult to be successful in mating and reproduction, but they can colonize anything. So there are trade-offs as a specialist and a generalist in terms of how they interact in the environment. So one thing we always point out is that no two waste streams are the same. This is really critical for understanding ecosystem function. Detritus of one animal is not detritus of another animal, etc. Same thing with plants. And understanding that process, understanding those differences is really important when we apply that in insects as food and feed sector. This is a paper published last year that just shows the degree of variability in protein, lipids, and nitrogen, and then the combination of the three. But you get this idea if you look at the substrates across the horizontal axis, is there's a high degree of variability, and not just across substrates, but within substrates. So this variation again can really impact mass production of an insect because depending on the resource, it's going to impact survivorship, development time, and downstream nutritional quality of what's being generated. So this past year we published a second edition of our Carrion Ecology book, and within it we have a chapter on insects as food and feed. And we talk about this idea of the circular economy or industry symbiosis is a new term that we hear a lot about, but this idea of waste production and how we can recycle it with black soldier fly, how we can integrate that with other industries, so the symbiosis part, and we can produce products of value, be it the larvae or the frass,
Tahir, when you
that can
can
be
add your current
used
trials to
downstream,
the table, please.
that can be used downstream for animal feed, but also other areas as well as bioenergy production, micronutrient production, etc. So again, when we think about insect farming, black soldier fly, yes, we recognize. Mealworm, we recognize. Cricket, etc. Macellaria, the blowfly, actually could be mass produced. It can feed on high protein diets and generate insect biomass quickly that's high in protein content. And there's been efforts in the past. I think most people are aware of like agroprotein's initial start with Chrysomia species, another blowfly. So there's been efforts in the past, but there are challenges with that as well. So with black soldier fly, obviously, it's neotropical realm, so it's from South America, Central America, Southern U.S. is now distributed throughout the world. Feeds on a lot of different things. Highly developed in the insect as food and feed sector, and It's getting a lot of recognition as a non-traditional model organism, which is beautiful because it's allowing us to bridge basic and applied research, which is creating opportunities for the insects as food and feed sector, but also beyond. So, with in terms of black soldier nutrition, it's really been focused on mass rearing ability, and this is just an example um, from Carol's work where you get an idea of where carbohydrate content and protein content impact the production, either development time, yield, pupil weight, etc., across. But you see that depending on the phenotype being studied, the nutritional profile changes. But this is from a protein carbohydrate perspective. And what I just want to remind you is to think smaller. What about those micronutrients? Because not all protein is equal. So when we think about the detritus example, that's one thing. Not all detritus is the same, but it's also from a vertebrate perspective. My lab, they give me a hard time because I always say carrion is not a hot dog. And what I mean by that is not homogeneous. We recognize that different parts of the animal have different nutritional profiles, which is going to impact the development of the insects that feed on them. So that nutritional variation is obviously really important because that's going to drive downstream phenotypic expression. Now, Macellaria, this is just a close-up of it, the secondary screwworm, and I think you'll appreciate why this work is really important in a minute too. But Macellaria is native throughout the southern United States. It's actually been found as far north as Canada, with change in environment, climate, etc. It is a primary colonizer. What that means is when the animal dies, it's one of the first insects to find it. It is relevant in my world of forensics because we often have forensic investigations where this animal may be present, association with human remains. Now, a lot of work historically on nutrition with these blowflies, be it Macellaria or these that are listed here, have really focused on like the digestive tract and what does it digest and how does it digest it, and where different micronutrients or resources are needed in production of this insect. And this is traditionally done for one primary reason, and that's for meiosis. And why is that important? If you haven't seen this in the news, the primary screwworm, which is the sister species of Macellaria, was eradicated in the 60s all the way down to Panama. And the reason being is that it's a massive impact on the livestock industry. It colonizes wounds of living animals and it will kill them. And think about it, it's not just about livestock. That is livestock, people, pets, etc. Well, screwworm is coming back. It's now up in Mexico. There's concerns it's going to come back into the United States. It's going to have a lot of detrimental effects on not just animal health, but also human health. Also encouraged these studies by Hobson, 1931. Wonderful, wonderful studies examining the physiology of these insects. Beautiful work. Encourage you all to take a look at them if you have a chance. So there is relevance with regards to the screwworm and why we need to understand the nutrition ecology of it as well. So for this work, we're really focusing on neonates. Neonates and mass production can be a really fragile part of the system. You hatch these larvae, and then you have to use them to mass produce, digest waste, etc. So it's the first feeding stage, usually less than 24 hours old. It's really small. We recognize that a larva can be 0.01 milligrams. The thing that we also recognize is that the nutritional requirements of a neonate is very different than what maybe a fourth instar black soldier fly larva needs. So understanding that vulnerable first stage of development is really important because I always like to say the colony is the heartbeat of the company. So if we can have a healthy colony that's mass producing neonates that survive to production and use, that can enhance the ability of the industry to be successful. And as mentioned here, very little has been done on this stage to this point in terms of like nutrition ecology. But if we can optimize this, we can make the system much better. So moving forward, we really wanted to look at how supplementing tryptophan and zinc reduce variation in the neonatal larval performance aspect, so the phenotype of the neonate. And we wanted to look at it from a generalist versus specialist perspective. So when we think about detritus versus carrion, again, both species have very specific roles where Hermetia lucens can colonize both, but Macellaria can only colonize vertebrate remains. So a lot of work has been done on both, but we don't know a lot about the nutrition ecology of the system. And so we wanted to do this to look at how phenotypic plasticity may be at play here. We weren't able to get at multiple populations, and that's unfortunate. We couldn't do that with this study. But future work would be looking at how different populations respond to these different environmental factors. So we can do G by E interactions and examine the phenotypes expressed. So first thing was we had to develop the methods, and we're working with these 15 mil Falcon tubes, and we were working with tryptophan, obviously, and working with zinc. And what we did is we doubled in the tryptophan and zinc in the diet, and I'll explain the diets in a minute. And we did it for both Macellaria and for elucens. So the objectives were to look at these two species and look at when we double tryptophan or double zinc or look at the combination, how that impacts these different parameters. And you may be saying, well, what's doubling and what's tripling? And I'll get to that in a minute. So also one thing to point out is we focused on the first 50% of the larval feeding stage, for 50% of the larval feeding stage. So we started with neonates and we calculated out using accumulated degree days, which I'll talk about in a minute, and said when's 50% of the development reached, hypothetically. So this is Macellaria, the colonies that we use. It's maintained at the lab, and this just shows you how we maintain in these cages. We collect the eggs, we grow them on beef liver, etc. Neonates for elucens, these are less than an hour old, and they're kept in a 100 mil mason jar with paper towel till hatch, so we could collect them fresh. Now, in terms of the diet, we worked with bovine liver for macellaria, so we could look up what the tryptophan or zinc concentrations were in that material, and then we could double it. And we also did that with the Gainesville diet, which is the standard, or I'd say baseline diet that we can use for mass production of black soldier fly. And we worked with Tyson Food Group to do this work. They analyzed all of our substrates to make sure that we were truly doubling the tryptophan or doubling the zinc in that material. So we would double it or double it, and we would place it in these mason jars. This is the liver smoothie that we created, and this is the Gainesville diet. And then we would put them into these Falcon tubes, and it would be with 20 neonates, 15 grams of diet uh, per tube for black soldier fly, and 3 grams of diet per tube for MAC or Macellaria. Uh, I didn't envy Erin having to work on the smoothie aspect of the liver, but that's like she did a great job. So we would count these neonates. They would go in these falcon tubes. They're covered so they could breathe, and then they'd be put on a slant in the growth chamber. And then Aaron monitored them for growth. This is the 50% point for black soldier flies. It was 7.7 days, and this is based on data that was generated by Jones et al. 2017 and Boatwright for Macellaria in 2010. So Macellaria was 3.25 days and BSF was 7.75 days based on an ADH calculation or accumulated degree day calculation. So then after that time point, we didn't want to have handling effects. So we put them in there, monitor, make sure everybody was happy. But then if we reached that time point, Erin would break them down. And what she would do is take and dump the contents, sort out the larvae, count them, survivorship, weigh them, etc. And then she would do length by weight with measurements using ImageJ software. And we also looked at feed conversion rates of the diet that was in there. And this is the basic stats that she used for analyzing the data. So what did she find? So here are the data that were generated, and what we find is that if we look at the control and the mean, we see that by adding tryptophan or adding zinc, we can increase the mass. And this is the coefficient of variation. And what's fascinating is if you look at the treatments versus the control, by doubling the tryptophan, zinc, or the combination, we could, in general, reduce the variation observed anywhere from 15 up to about 30%. And that's for mass. On survivorship, it really was dependent. You know, if we added tryptophan, we could reduce the variation associated with it. Same thing with a combination. And why is that important? Because when we look at this in general, you would say, well, there was no impact. I mean, if we're looking at survivorship or mass, there's no impact. But the variation within the sample was impacted. The same thing when we look at feed conversion rates, we could see that we could reduce the variation quite a bit just by manipulating these micronutrients. When we look at mass malaria, a similar response for mass, but the similar response. What I mean by that is, same data were recorded, but we saw the opposite. When we started manipulating these micronutrients, we actually increased the variation observed for mass. But on survivorship, the combination had the greatest impact. It really reduced the variation observed. And then for the feed conversion rates, again, we see that there was an increase in variation. So what it shows you is that the specialist responds to these influxes of micronutrients different than the generalist. And that may be in relations to their biology. One is more flexible in response versus the other one, which is more specialized. So composition can change the rate of decomposition. We know that. And how an insect utilizes these nutrients is going to be really important for understanding these life stages and how they respond. The generalist, all treatments reduced the variation, but it was most efficiently utilized in excess nutrients. So there's more plasticity in that system. Where the specialist had reduced across, uh, for tryptophan, had reduction across the board, but zinc in the combination had less plasticity. So it was really nutrient specific in terms of what outcome was observed. So C. macellaria, one thing that's fascinating about it in terms of its response, because we say, well, zinc increased the variation, is that actually the adult accumulates a lot of these metals in the environment rather than the larva. So where we know macellaria can go out and feed in the environment and actively pursues looking for nutrients, and we know that elucence doesn't necessarily have to do it. It will, but it can rely on its fat body to be productive. It shows this deviation in these decomposers in terms of where their niche is and how they operate within that given ecosystem. So the next part of the study was really looking at, okay, if we supplement amino acids, so we're moving away from just tryptophan, and we're saying, well, what about all the amino acids? So if we supplement the amino acids, how does Hermetia lucens respond? So I'm going to focus more on it. How does it respond to these supplementations? And the thing is, this isn't new. We know that this is done across the board for most livestock or with pets, and that is finding optimal nutrition for maintaining the health of your pet, but also for mass production. So what's being done in terms of this work, it has been done before, but for other species, we just have to apply it here. So we're looking at how we can apply this to optimize or find that bottleneck in mass production of black soldier flies from an amino acid perspective. And what we think is that this could help really enhance the colony production aspect. This is done if you think about poultry with the hub and spoke model, they have the nurseries where they rear these chicks and then they're provided a specific diet in broiler facilities or if they're in layer facilities or breeder facilities. And the same is done with dairy cattle, beef cattle, etc. And a lot of work's been looked at in terms of mass production of the insect and how to formulate diets to produce them. But again, it's really focused on like protein, carbohydrates, and lipids, not as specific as what we wanted to focus on. So with this study, what we did is we looked at doubling and tripling the essential amino acids in the diet using Gainesville as the baseline. And then we also did it for the non-essential amino acids as well. And we looked across the board at the impact at these various phenotypes. And again, we looked at the 50 during the first 50% of the larval feeding stage. So not the full scale. It's very micro scale, and only half of the development time. So this is our colony. This is the old cage. We got a new fancy metal cage now. It's much better looking in our greenhouse, but we can collect our eggs and we maintain it right here at A&M. It's the Shepherd strain that originated in '98 out of Alma, Georgia. So method recap, again, Tyson made sure that we were getting the right nutrients at the right concentrations so that we could precisely double those essential amino acids or triple them in solution. Same thing for the non-essential amino acids. Then we could apply it to the Gainesville diet, place them in these falcon tubes, and then we could monitor them in this incubator. Again, that's the 50% point for larval development, 7.75 days. After that time, each tube was broken down, measurements were made, survivorship, feed conversion rates, etc. Statistics, very much like the others. So what you look when you look at this graph, this is mass of the larvae. Across the horizontal axis, D is double, T is triple, and this is for these amino acids, and the Gainesville controls at the end. What's fascinating is this is what the mean is for the control. So this shows you when we add double or triple of amino acids, which ones were doing better, and how it impacted the variation in the system. And what's really fascinating is methionine. I love tryptophan. I think it's a fascinating amino acid in terms of microbe insect interaction or micro host interaction, interchemical communication. But methionine, we all recognize as an essential amino acid, is really important. When we doubled methionine, not only did we get a 50% increase in larval size, but we also got almost a 50% reduction in variation. Same thing when we looked at the remaining amino acids. This is the mean. When we added the non-essentials, you see that we get this increase in mass. And for many of them, well, all of them across the board, but many of them, substantial reduction in variation. This is feed conversion rates, and this is just the same thing with your amino acids across the horizontal axis, double and triple. And this is the variation in feed conversion rates. That's the mean. And what we see is that across the board, that it was really dependent on the amino acid in terms of the effect on feed conversion rates. It doesn't look like it had much of an effect, but it did on the variation. When we see it for the remaining amino acids, very little impact in terms of the phenotype being expressed, but the variation expressed was really important. So this is just an example of sometimes we target a certain phenotype, but we forget about some of these underlying characteristics of that phenotype, such as the variation. So again, if we can reduce the variation of production 50, 60%, that could have a massive impact on the business model being developed in terms of what can be produced and when for a given client. So what did we find for the FCRs? 12 essential amino acids and the seven non-essential treatments decreased the feed conversion rates. Not the best, but it did decrease the variation. Um, Let me rephrase that. It decreased the feed conversion rates. It made it better. I should not have put decrease. It improved the efficiency of the system. With the size of the larva, almost every amino acid increased the size of the larva and lowered the conversion rates. With weight, the biggest impact, methionine. Uh, it's Methionine is in most animal production systems is a major limiting nutrient. It's the same thing for black soldier fly as well. So we need to think about how we can supplement methionine to enhance black soldier fly production. Now, this is all done at micro scale in the lab. So obviously the next step is like, well, can we take it to industrial scale? So as I mentioned, methionine is supplemented in a lot of different animals at low rates. So with swine, we know it's done, and it not only enhances the health of the animal, but survivorship, etc. The same thing with poultry. It improves growth and survivorship as well. So again, not reinventing the wheel, just verifying the wheel on the insect side. Now that led Aaron and I to talk a lot about, well, what's the value of decrease in variation? And I talked to David Zilberman at Berkeley quite a bit about this. He's an economist, and we talk about, well, what does this mean? So this is not David's assessment. This is Jeff Tomberlin and Aaron Harris. But if we look at doubling methionine, we get a 55% increase in weight and 45% reduction in the variation expressed. What does that mean in terms of value? So we published a paper a number of years ago, and we talked about, well, if we had a 0.6% increase in production in a 20-ton facility, that could equate to a $250,000 increase in gross gains. So if we can increase it by 55%, that could have a massive impact. And we're focused on the chicken treat market, which is a specialized treat market in the US. I'm not sure if it's in Europe, but people will pay up to, this is $17 per kilogram. That is wholesale. Um, if you were to go to a store, you would pay double that. So the profit margin's massive. So if we apply these gains just to that market, well, it's quite substantial. I mean, it could be a massive increase of production that could feed into this industry, create revenue. And if we do it for a 100-ton-a-day facility, it could be quite substantial. It was a fun exercise, but it gives you an idea. Uh, and as Aaron pointed out, it's not taking anything else into account, demand, production, cost, labor, etc. It's just saying in general, this is what it could look like. I keep hitting the wrong button, sorry. Now, if we take it to broilers or poultry production, where feed is much less expensive, it could still have a major impact, and that's something to consider. Now, is the broiler market what we're going to go after? Probably not, because replacing 1% of the broiler feed is astronomical in terms of what would demand would be. But if you look at like breeders, much smaller market, this is actually achievable. So there may be an opportunity to explore how to increase the efficiency, decrease the variability, better predict the production, and then engage the poultry industry and say, hey, we can work at supplying this material. We can predict better what our production will be. So natural versus synthetic, maybe this is where black soldier fly comes in as a value downstream, and reflection on this work is if we can enhance methionine concentrations in black soldier fly, maybe there's value there for natural production system for these other industries. It's not just about increasing the value of the production efficiency of the black soldier fly, but also generating methionine that can be extracted and utilized in other sectors such as the poultry industry. So we have to think about how to do this sustainably. Go back to that phrase, the industry symbiosis. How do we work with these industries to produce value for the insecticide food industry? Now, all of that work has been done at micro scale. So where are we? Where are we now? So I'm just showing this picture. I had a visitor from Italy, Asia Zanzat. She was from the University of Perugia, and we looked at how in supplementing zinc or tryptophan or some of these other essential amino acids at an industrial scale. When I say industrial scale, we're talking 8 kilograms of diet, 10,000 larvae. How does it impact the growth of these insects, and how does it impact downstream egg production? And why do I say that? Because zinc is important for egg production in insects. So, can we supplement zinc, generate adults that are better at mating and producing fertile eggs? So, we're going down that path. But that in combination with Tara McNeil, another PhD student in the lab, is how does it impact thermal responses? We're looking at heat stress. These pans can get up to 40 plus degrees Celsius when mass producing. If we supplement zinc, which is important in thermal stress responses, can we get better conversion rates at these high temperatures? Can we get better survivorship, etc.? Then what we can learn from this is if these pans get to 40 plus degrees, it's not as detrimental on the system as it would be in the absence of the zinc. So with that, I want to thank you for your time. I'm here to take questions. Thank you.